Biomedical Sciences in the Charles Schmidt College of Medicine at Florida Atlantic University, and the founding executive director of the FAU Brain Institute. Prior to his arrival at FAU, Blekely held the Allan D. Bass Chair in Pharmacology and Psychiatry at the Vanderbilt University School of Medicine for 21 years. And when appointed, was the youngest to hold an endowed chair in university history. At Vanderbilt, he directed the Center for Molecular Neuroscience, the Vanderbilt NIMH Silvio O Conte Center for Neuroscience Research, and the Vanderbilt NIMH Postdoctoral Training Program in Functional Neurogenomics. In 1998, Dr. Blekely and Dr. Elaine Sanders-Bush launched the Vanderbilt Brain Institute and generated a nationally prominent PhD program in neuroscience. He holds a dozen patents for his gene discoveries, novel research models, and potential therapeutic interventions, and continues to strive to translate his fundamental research to novel therapeutics. He has served on multiple advisory panels and supported medication development for multiple companies. Welcome, Dr. Blekely. You can take it away.
Thank you, Pramila. Very nice introduction. I'm going to share my talk. Hopefully you can see that now.
Yes.
Thanks so much for the introduction and for welcoming me into the Brain Foundation and supporting our research. So today, I'd like to continue a bit from a talk I gave last year that some of you may have seen when I talked about a mouse model that shows ASD-like phenotypes and how we could ameliorate those, thinking about immune signaling. And so today I'm going to extend this a bit and tell you where we've gone with this work and how this may point to bidirectional interactions between serotonin and the immune system. Let's see, can I advance this? Of course not.
I think you should click on the slide. This is some new update. If you click on the slide, it might.
Okay, great, thanks. Many people know about serotonin signaling, and I'm not going to spend a lot of time talking about the anatomy of it, except at one small place in the talk. But most people know about it because serotonin signaling can be potentiated by the use of antidepressant molecules that target the serotonin transporter. These are drugs such as Prozac and Lexapro and so forth. And early in my work, this involved cloning the gene, encoding the serotonin transporter, and working out fundamental mechanisms as to how antidepressants interact with this protein and how it's regulated. But a good bit of our work moved over into the autism space when we found that by in situ hybridization, when we looked at the expression throughout development, we were surprised to see the serotonin transporter mRNA expressed very highly in a mid-gestation mouse embryo. So this is middle of embryonic development for a mouse. And it suggested to us that maybe if there were deficits in the function or expression of this protein, this might lead to some developmental abnormalities. And certainly we know that serotonin plays a role in many behaviors, and therefore maybe a neurobehavioral disorder as well. Now, thinking about serotonin at the time, what I knew about serotonin in autism was hyperserotonemia. This had been a biomarker that had been described 60 years ago as of this year by Shane and Friedman, indicating an increase in about 25% of individuals with an autism diagnosis, showing as shown on the right panel in orange, and just one of many examples, a significant group of subjects who have this elevated amount of serotonin in their blood. And this serotonin that's in the blood and that's accounting for hyperserotonemia is coming from platelets. And platelets store 99% of the serotonin in the blood. And this serotonin is made in the gut. It is picked up in the circulation by platelets through the protein that I was mentioning before, this serotonin transporter, this antidepressant sensitive serotonin transporter. And the platelets express this protein. It's accumulated inside the platelets and stabilized there in secretory granules, and they circulate around the bloodstream and use that serotonin in various ways. Serotonin levels in the bloodstream are highly heritable, more so than height. And we know that they're influenced by both genetic variation in the transporter, as well as SERT, interacting proteins, and other 5-HT homeostatic genes. So given that SERT was one of the proteins that could control blood serotonin levels, steady state levels, because of packaging it into these platelets, we wondered whether or not there might be some genetic changes in individuals with autism in SERT itself. And so with Jim Sutcliffe back in the in 2005, we published a study looking for rare genetic variation in multiplex families with ASD. And we found 5 coding variants, which was, these coding variants are very rare. And in the number of subjects we looked at, it was quite striking to find this many. What was even more striking about these variants is that all of them, I'm just showing 4 here, all of them did not eliminate the function or reduce it. They actually increased the function of the transporter. They made it hyperfunctional. That was something very new to me. And it suggested that there might be a contribution being made, at least in the individuals with these variants, in traits of autism. And we showed that in the report that the most common of these is Gly56-alanine variant at the top that you see there, tracked with rigid compulsive traits and sensory aversion. So we were quite interested to see if this in vitro phenotype, which was replicated in the subject's own lymphoblasts, would carry through if we expressed it in mice in vivo. And before I get to the mice, just let me segue for a second and show you the model that we have for this increase in SERT activity. And I show here just induced by ALA56, the variant that we've studied the most there on the right. SERT star is an increased activity state of the transporter. And we know that you can move between the wild type and normal activity of SERT to the SERT star conformation via the activation of an enzyme called P38 alpha MAP kinase. This kinase is a well-known target of stress sensitive, of stress, cellular stress and systemic stress. And one thing that can activate that pathway is the inflammatory cytokine IL-1 beta. And we showed that that cytokine would stimulate SERT activity in cells and in ex vivo in brain preparations. So one idea was that ALA56 was inducing a state permanently that IL-1 beta induced in SERT transiently. And so how might that play out then in terms of behavior or in terms of physiology? And that's why we made a mouse expressing that variant. So in that mouse, what we showed on the left was that these mice were exhibited hyperserotonemia. At the time, this was the first mouse model that had replicated this blood biomarker in ASD. And we were very excited just with that, and probably just should have stopped and then, published it. But we had to do a few more things to look at the physiology and behavior. And one important thing that you see on the right is that when you pulse serotonin into the brain and you measure how fast it clears from away from the pipette where you puffed it, which is the descending limb of this curve shown here, you see that the Gly56 alanine, the wild type expressing mice, clear serotonin at a nice rate, but the ALA56 mice clear serotonin much faster, indicating that in fact the transporters in vivo, because the density of the transporters was normal, is likely to be in this cert star state, clearing serotonin away from its receptors faster and more efficiently than a normal system should. We also found that the G56A or ALA56 expressing mice, their serotonin neurons also did not fire at the same rate as the wild type cert expressing neurons, indicating that there were two mechanisms by which diminished serotonin signaling could occur, one via diminished excitation induced release and the other by faster clearance by the transporter. Now at the behavioral level, I don't have time to go through the number of behaviors we studied, but I just wanted to show you one, which is in this simple test called the tube test, which is a test in some studies where you've established dominance hierarchies as a test of social dominance. In this case, these are not animals that have been in those kinds of hierarchies. And what it really queries is social avoidance. And what you see in the plot below is that when you plot the number of wins or losses by animals entering the tube and meeting in the middle, and a win means that they exit the tube nose first, essentially either pushing the other mouse out or the other mouse backs out. The ALA56 mice back out, and they show this pattern of social avoidance. Now, if you manipulate that regulatory pathway that I described, this P38 alpha MAP kinase pathway, what you can do is actually normalize many of the behaviors that we show in our work. And I'll just show you one piece of physiology, which on the left you've already seen. That's the elevated clearance rate for serotonin in the cert ALA56 mouse. If you treat animals with this P38 MAP kinase inhibitor, it's called MW150, you see that the rates of clearance are essentially the same. So this is a 1-week treatment once a day with this P38 inhibitor, and they tolerate this drug very, very well. Additionally, the social behavior deficit that I described is normalized. So the purple bar is just wild types interacting with each other, showing 50-50 wins for whoever we put on one side versus the other. And the ALA56 animals, again, do not win in this test. But when you give them MW150, these animals show a normal win frequency. It looks actually like they're doing a little bit better here, but on average, that didn't occur. It was essentially normalized. So now I want to get back, I want to move away from the animals for just a second and just remind you that I was speaking about this P38 MAP kinase pathway that puts the transporter in its hyperactive state, and that you could activate that pathway with IL-1 beta and IL-1 receptors. And we showed that we could trigger peripheral inflammation and get this change to occur in the brain within 30 minutes. And it's mediated by IL-1 beta in the brain. So we were interested to find at about the time that we identified our variant and began studying it, that in fact the IL-1 receptor pathway and P38 MAP kinase pathway were shown to be elevated at the transcript level in microarray analysis of temporal cortex by Karoli Mernix and Tony Persico. So this was very exciting to us that, in fact, we had walked our way into an immune regulatory pathway that seemed to be upregulated, and inflammatory markers have been reported many times now in association with autism, either peripheral or centrally. And so we've been interested in this conversation going on possibly between the immune system and serotonin signaling, less from a mechanistic perspective, and perhaps that there's something endogenous is happening here that might relate to autism. So one thing that we did was, as I said, we created a peripheral inflammatory insult using LPS and asked whether or not serotonin clearance in the brain is accelerated. And on the left panels, you can see that if we inject saline, the curves pre and post saline don't change. But if you inject LPS, which will give us an increase in IL-1 beta in the brain, you see this shift toward this steeper curve and faster clearance, and it's quantified on the right. This change in SERT-mediated clearance is paralleled by changes in serotonin uptake if you measure tissue ex vivo, and that's dependent on the IL-1 receptor. So we believe the pathway that I just showed you in terms of activating SERT is in the middle of this ability of the serotonin system to respond to LPS. And here are those synaptosome studies. I'll skip on through this, but again, identifying this IL-1 receptor and IL-1 beta system, regulating the transport as an endogenous pathway by which serotonin neurons can communicate peripheral inflammatory status to changes in behavior. So now, are there IL-1 receptors on serotonin neurons? Well, yes, there are. Here's an immunolabeling approach where we're looking at in the leftmost panel serotonin neurons in an area called the dorsal raphe that are stained for serotonin, and in the middle panel you see a stain that's highlighting the IL-1 receptor. And you can see when we overlay them, there's quite nice correspondence, particularly in the dorsal areas of the dorsal raphe. This dorsal lateral area is very rich in IL-1 receptors and one of the richest areas in the CNS. So then one question became, what happens, what do these neurons actually do? What are their targets? And might that tell us something about behavior? And this has been an activity that Paula Kurtzel in the lab has been working on for the last several years. And we're beginning to make some headway. We use a technique called CFOS labeling. CFOS is a protein that's induced by neural activity, and CFOS positive cells can be counted to indicate how many of cells in a particular brain region are activated. Now, one question that we've had is these neurons on the left, these neurons in the dorsal lateral dorsal raphe nucleus that are serotonin positive and positive for IL-1 receptors, where do they go? Where do these neurons go? And can we show that they change the activity of those neurons? Now, one way to do that, we can't be following all serotonin neurons. Only ones we want are the ones that are IL-1 receptor positive. And to do that, we've used a genetic approach where we've eliminated the IL-1 receptor, which you see on the far right. So this panel just demonstrates the red spots indicate that these neurons in a mouse, we've eliminated the IL-1 receptor just on the serotonin neurons. So now what we can do is give LPS peripherally, look centrally, and ask which neurons are responding to LPS and changing the activity of targets that these neurons project to. Now, this is a lot of data here, and I'm not going to go through all of it, but there's really just two bars to pay attention to. We've queried males and females at different brain regions for CFOS positive cells. And what you can see, let's focus on the females in pink there. You see that when we give LPS in the periphery, this region of the brain called the lateral habenula, you see fewer of these positive cells, indicating that this nucleus has been inhibited. And when we go over to the bars, the two bars to the right, these are those neurons where we've taken out the IL-1 receptor. And you can see that this effect is normalized. So there is an inhibition of lateral habenula neurons in females that is totally dependent on IL-1 receptors in serotonin neurons and no others. The lateral habenula of a male, on the other hand, we see stimulation of the activity in the lateral habenula, and that is normalized by IL-1 receptors, a striking sexual dimorphism that is underlying behavioral responses to peripheral inflammation, now acting on the brain on a discrete pathway overseen by the lateral habenula. And why do we care about that? Well, the lateral habenula has been implicated in the pathophysiology of autism. I just want to point one recent paper out from Gurman et al. That used magnetic resonance imaging to show that there's a significant increase in the volume of the habenula in individuals with autism. The lateral habenula is pretty interesting because it mediates reward behavior, and particularly aversive behavior to either lack of reward, but also sensory stimuli that are aversive and social stimuli that are aversive. It also contributes to behavioral flexibility and inhibition, some cognitive states, sleep cycles, and pain responses. So a number of these are characteristic responses of a peripheral inflammatory insult, but they're also seen in individuals with autism as well. Now, I've tried to tell you about how serotonin and its modulation, either the neurons or the transporter by immune status, can change behavior. If we know something about it, we can tackle it and see if we can normalize the physiology and behavior that's disrupted. But one other aspect of this is that serotonin, this is not just a one-way street. Serotonin neurons also regulate the brain's immune system. And so here what I show is just a little illustration from sorry, that's not spelled right. It's Ho et al., 2013. Neurons release a number of molecules that regulate the status of glial cells, and in particular microglia. It turns out serotonin suppresses microglial activation, which is a pro-inflammatory state, and keeps microglia in a state that's supporting normal neural health and synaptic function, synaptic plasticity, through such as secreting TRAC beta to regulate neural precursors in the hippocampus, which we know is makes a contribution for antidepressant-induced antidepressant activity. So if we're not releasing as much serotonin in the case of the ALA56 mice, if we have hyperclearance of serotonin as shown with the ALA56 mice, if any way in which we're diminishing our serotonergic output, this is going to lead to microglial activation because of the lack of inhibition. One way we could test that is to go back to our mice that have the ALA56 mutation and do a little sampling, which is something we've just begun to do now with Brain Foundation help. And what we see is just this 1 point mutation. In fact, it's a very, very subtle change in the structure of this transporter, but just that alone is enough to increase inflammatory cytokine expression. Here you see IL-1 beta and IL-6. You get an IL-10 response as well. And this is now CERT ALA56 mice, the mutant mice that I showed you before. And this is in the midbrain. We see it in other brain regions as well. But another interesting aspect of this is that we see it in the periphery. And so here we're looking at the spleen, and you see also inflammatory cytokines elevated. You also see enlargement of the spleen in these mice as well that's genotype dependent. So what I think is happening in this animal is a case where serotonin neurons are clearly built to respond to inflammation. They respond to it early in development, they respond throughout life, changes behavior, and the changes in serotonin signaling capacity also change the immune system. That's been known for quite some time. But now with this point mutation in one protein, we're a little cleaner in our assessment that, in fact, that one output of these serotonin neurons is sufficient to do what I showed you in the brain. This, the source of this effect, of this serotonin effect and SERT effect in the periphery, may well have to do with the enteric nervous system, which has serotonin neurons, or other aspects of peripheral serotonin that we haven't begun to investigate. And that's part of what we'll be doing in our new studies with Brain Foundation support. So let me just wrap up now and give you a summary. So I've told you that serotonin is expressed early in brain development, and that's led us to want to understand how alterations in serotonin homeostasis contribute to neurodevelopmental disorders. Along the path of that, we identified rare SERT mutations, and these are functional. They induce changes in SERT activity, increases in SERT activity that are seen with the endogenous transporter when we make mice, and that they're similar to what you get if you treat preparations with IL-1 beta. We look at this in transgenic mice carrying that variant, and we see changes in physiology and reversible ASD-like behaviors. We know that high IL-1 receptor expression in brain serotonin neurons leads to the change in activity of a few other brain nuclei I haven't talked about, one being the lateral habenula, and that SERT hyperfunction on its own, without any other stimulation, induces a pro-inflammatory state in the brain and periphery. And it really suggests this bidirectional conversation going on between serotonin and the immune system as a normal aspect of physiology of the brain and periphery, but is disrupted when you diminish serotonin output. And we just showed you one way to do it by changing SERT activity. So with that, I'll stop sharing and see if we have any questions. Oh there we go. Okay. So thanks a lot. And let's see, I am going to go over to the Q&A and see if I see any questions.
There's one question.
Does SSRI reverse the behaviors? We haven't done a lot with SSRIs in the model. They would be predicted to do so. And in some work that Jeremy Veenstra Vanderwill has done with these animals, some of these behaviors, yes, can be normalized with SSRIs. We haven't done this extensively. It would be good to do so. One of the problems, however, in using SSRIs is that no doubt many of these serotonergic pathways are not, they're not things from a clinical perspective. We don't really want to block them all. We'd like to get to the ones that are in a hyperactive state. Now, remember, these mutations I showed you are rare. So thinking more broadly, it'd be better if we could go after ones that are hyperactivated. And I suspect that these would be because of immune activation, pro-inflammatory activation.
Can I ask a question?
You certainly can.
Yeah, I'm a clinical immunologist, so I was wondering, what's the serotonin uptake stat of the congenital deficiency of the some of the genes, which is usually causing, like autoinflammatory syndrome, causing the over-production of the IL-1 beta, or the deficiency of the IL-1 receptor antagonist. Those are very typical autoinflammatory syndrome. What happens in these patients in your serotonin pathway?
Yeah, I think that's a really interesting idea. You know, the most that one typically sees in the literature is related to mood disorders. and depression and high frequency of mood disorders in subjects who have chronic inflammatory signaling. And some of these occur during during adulthood. When this occurs early in development, I think that there'd be other changes associated with behavior that I would predict would have a serotonergic component to them.
Mm-hmm. Yeah, because the very difficult patient and like OMID type patient as very sick, very irritable, like a serious meningitis type symptoms. So I was wondering, but I'm not aware of any of the report with a serotonin mechanism in these patients.
Yeah, and when they are reported, like I said, they most often have been evaluated in the context of mood disorders, certainly irritability, anxiety, those kinds of things. But the kinds of social behavior changes that we see in our animals, we also see repetitive behavior in them as well. And we see poor communication between pups and mother as another hallmark of their behavioral changes that is seen in a lot of animal models of ASD.
So the treatment we use anakinra, that is like a human recombinant iron receptor antagonist. Does it work? That is human, but I think the mouse iron receptor antagonist, I think it's available. Yeah, I wasn't sure. I'm curious if it's available.
We haven't tried that, but we have genetically eliminated the IL-1 receptor and done that just in serotonin neurons. And it definitely prevents the ability of a peripheral immune insult to affect CNS serotonergic signaling. But in terms of the ALA56 case, we haven't looked yet.
Okay.
So let's see, I have a question about how prevalent are the certain mutations in the general population? Is there evidence for increased generalized in humans with certain mutations? So the ALA56 variant that we worked on is the most common one, and it's at about 1% or less. And so, there just has not been an attempt to try to gather enough of a population to do, those kinds of experiments. It would be great. The other mutations I talked about are, private ones, essentially. They're in individual families. And then the follow-up, what is the mediator between serotonin and inflammation? How does it talk to the immune system? Why use serotonin for modulation? So there are serotonin receptors on microglial cells, and our current hypothesis is in fact that, neurons talk to microglia through many different signaling molecules I showed you, and serotonin being one of them. There's a lot of interest in how SSRIs may reduce inflammation in relation to neurodegenerative diseases. That's caught the attention more, I think, than people looking into autism or in depression. But I think it's quite an interesting dimension, and we're trying to push this for funding from the NIH. Why use serotonin for modulation? I'll wave my hand and think teleologically here, but I think that, the kinds of things that need to happen, and this has been said by psychoneuroimmunologists for a long, long time, is that you need certain behavioral states are protective in the context of inflammation or infection. And the things that you need to change by way of your behavior, your metabolism, and your physiology are all under the control of serotonin signaling. And the serotonin system is very broad, and it can control many different behaviors in parallel. And so one important outpost are these neurons to affect a number of different behaviors, like stop eating, get away from people so that you don't infect the rest of everybody else around you. Get some sleep, conserve your metabolism. All of these things have serotonergic modulation available and use. And I think the immune system, you know, we have to think about the peripheral immune system as going after discrete circuits to change behavior, not just to go in and make people sick. So I think we're at time. I guess I can move on to our next speaker in the session, who needs no introduction, so I won't give him any. No, he's my friend, Dr. Aurelio Galli. He's director of the Gastrointestinal Biology Research in the Department of Surgery, and also holds an appointment in the Department of Neuroscience. Previously, I got to know Aurelio because together we were on the faculty at Vanderbilt. He was in molecular physiology and I was in pharmacology.